Om Swastiastu. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, all. And now I would like to introduce myself first. My name is Ida Putu Suryanti, and I will share you about the results of our research. And the title is the additions of a local microorganism to the densities of bacteria and macronutrients for the catfish Clarias SP Biofloc system waste. And this is the backgrounds of our research. The cultivations of catfish, Clarias sp, is the choice of some people during the COVID-19. And the popular catfish farming methods is called the Biofloc systems. Biofloc system has several advantages, including being able to be applied in a closed place, saving land, water, and feed more efficiently. And water waste from catfish farming with an intensive system contains macronutrients needed by plants. One of the group that developed it is a Additon. This is the fish cultivations group, and the name is Ulam Mandiri. The location is Temukus Village, Banjar, Buleleng, Regency, Bali. And this is the community group that have been cultivating catfish using biofloc systems. The increased the density of bacteria can help the degradation process and nutrients levels. So we need microorganisms like a local microorganism to be a starters for this. And the densities of bacteria and measuring the levels of the macronutrients content in the waste of Biofloc system of catfish for liquid organic fertilizers in the futures is the one things that are focused today. And this is the research of the methods. First is the preparation stage that for the preparation sample. And after that, the productions of the local microorganism. And then implementation stage is additions of each concentrations of microorganisms. It means the local microorganism for the varieties of the concentrations start from 10% to the 50% into the Biofloc wastewater. And the control, we use the effective microorganism for products. And after that, we are measuring for densities of bacteria with the methods, namely a total plate count. And after that, a local nutrients content testing stage to know about the contents of the carbons and the nitrogens, phosphorus, and also the calcium. And after all of this, we are going to analysis of the data, and this research will be analyzed with the quantitative approach. Macronutrient data will be presented systematically in the forms of the tables and graphs, and after that bacterial density data in colony forming units per milliliter will be analyzed using statistics. And this is our results. The table one told us about the numbers of the bacterial colony in each Biofloc pool for the catfish cultivations, and showed that the numbers of the colony in all treatments is varied greatly. This indicated that the additions of the local organisms as a starter affects the numbers of bacteria present in the Biofloc system. And the highest bacterial colony density was found in the treatment tree. This is 30%, and the number is 16 times 10 to the powers of 7 colony forming units. And the next is based on the results of the photosynthetics test. The hypothetic test explained that a significant, it means that the variations of additions of microorganisms, the local microorganism with various concentration can have the effects on the numbers of the bacteria in each samples. And the results of the further test can be explained that the numbers of the bacteria in each sample was not significant compared to the control. Yeah, and so for density of bacteria, same as with this product, it means that effective microorganism 4. And this is the table 2, show us about the content of the macronutrients from catfish waste in a Biofloc cultivation system with the additions of the various concentrations of microorganism. And it can be seen that the contents of the macroorganic nutrients, organic matters, nitrogen, phosphorus, and kalium show the very varying results. The highest organics carbons content was found in a catfish Biofloc waste with the additions of the treatment 4, yeah, the concentration of 40%, and namely 0.271%. The contents of organic matters, nitrogen, phosphorus, and kalium in the same treatment also showed the largest amount in this research. And this is the conclusions of our research. The first is showed that the additions of the local microorganisms, it means that the concentrations for the 30% in a treatment tree gave the highest numbers of bacterial density. And the namely is 16 times 10 to power 7 coloniforming units per milliliters. The additions of variations in the concentrations of local microorganisms affected the density of bacteria. And the results of the further tests stated that there was no significant difference between the density of bacteria seen from the numbers of the colonies and the control. And the second, the highest micronutrients were found in the treatment E4 additions of the local microorganisms at the concentrations of 40%. Successively, such as C organics and the nitrogen, phosphorus, and also the kalium. In the ends of the goal will be to make it as a liquid organic fertilizers. Okay, that's all about our presentation today, and I hope this is useful for us, and I will say thank you very much, and Om Santi 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 Om, and good afternoon.